Thank you to everyone for joining us virtually for our summer science series. I'm Heidi Sauerwein, and I'm the program coordinator with the Indian River Lakes Conservancy. We are missing you in person tonight. Normally we hold our series at the Redwood Hill Preserve in our beautiful trailside learning classroom, which you can see in this picture was getting a new coat of stain earlier this summer. And our little owl face there. If you've never been out to the Trailside Learning Center in the Redwood Hill Preserve, we invite you to come in Redwood, New York. And you can also sign on our website to receive emails about events that happen out there all throughout the summer and fall normally. So I would like to say a little bit about the Indian River Lakes Conservancy. We are a donor-supported land trust. We are dedicated to preserving the unique natural character of the Indian River Lakes region. And so I put a couple of maps up here in case you're not familiar. One showing the county that most of the watershed's located in. And then this is a map of our watershed. Okay, our mission at the IRLC is fourfold. It encompasses protection, which is conservation of land and water, education, recreation, and we do it forever, or we have the long range view of things. Tonight's presenter is Kate Cleary. She is a SUNY Potsdam Environmental Studies professor. And I'm going to give a little bit about Kate in just a minute. We hope that you also join us for our next presenter coming up on August 27th. Nina Schock, she's with the Adirondack Loon Center, and she's been a presenter, one of our favorite presenters for our series just about every other summer. So we hope you can join us then. So a little bit more about Kate. Kate is currently an assistant professor in the Environmental Studies Department at SUNY Potsdam. She is a conservation biologist with experience working in diverse ecosystems in the Rocky Mountain West, Latin America, and most recently, the Adirondacks. She holds a PhD in ecology and conservation genetics from the University of Idaho and a master's in conservation biology from Colorado State University. In between earning these degrees, she served as a Peace Corps volunteer in Guatemala from 2007 to 2010 and conducted research as a Fulbright student scholar in Costa Rica in 2013. Her current research interests are relevant in both the tropics and the North Country, as she is interested in conservation on private lands, wildlife habitat, connectivity, and sustainable and biodiversity-friendly agricultural systems. She moved to the North Country 4 years ago with her family and her two little twins and loves the tight-knit community here, the amazing local food movement, and being right next to Adirondack Park. So, oh, and a little more about logistics. Asking a question, you can use the chat box or you can unmute yourself and ask a question as well. And if you use the chat box, I can go ahead and say it out loud to Kate. So please engage with her. She's normally a professor, so she's happy to answer any questions or hear any comments from you along the way. So without further ado, we welcome Kate.
Okay. Hi, everybody. Can you hear me okay?
Yes.
Okay, great. So thank you, Heidi, for that introduction. And I will say that, yes, I am a professor, but I've only been teaching online since COVID hit, right? But this is our reality. So this is actually great practice for me. So I will be keep an eye on your hands up on the chat so that I can answer your questions as they come up. Because as Heidi said, I want this to be like interactive. You can ask a question at any time. You don't have to wait till the end. So thank you all for coming on a beautiful night. And from many different places I see in the chat, at least two people from Canada, which is great because my talk is about a region that spans Canada, and the US. So I'm going to go ahead and start sharing my screen with you to show you. Okay. So as you probably know from looking at the announcement for this talk, I'm going to talk to you today about the movement of wildlife populations. And so initially when I proposed this talk, I was going to talk about how genetics can help measure wildlife movement, but just was a little bit dry. And so I wanted to make it just a little more fun about what we know in this area about how wildlife is moving and just some studies that have been done. Because a lot of times if you're not a scientist or a research person or professor, you don't have access to the databases where you might see this literature. So I'm going to share with you guys some of the cool work that's being done by other people in this region on wildlife movement. And then I want to hear at the end what wildlife you've seen moving through your yards. And obviously, really squirrels, that's not very interesting. But if you've seen a moose in your yard or you think you had a wolf sighting, save that for the end because I want to know about those too. Okay, so just to start off my talk, when I was a little kid, this was like my vision for my life was protecting spaces like this picture. This is from Southern Ontario. And I thought that I was just going to be out there all alone with not a human in sight, like trekking through the forest and discovering unknown animals. But then by the time I got old enough to actually do this, it actually looks like this, right? There's places that look like this. But a lot of the Earth's surface looks like this now. So I quickly came to realize you can't protect animals without thinking about how they're going to move through places that humans use, right? It's just not possible because there's not enough places left on Earth that humans don't use. And I don't know how many people are familiar with this statistic, but actually, so as Heidi said, I work with agriculture and biodiversity, and about 40% of the Earth has already been converted to agricultural uses, and that's crops and ranching, so cattle or other livestock grazing. That's 40% of the terrestrial surface. So this is a statistic that I don't know if everyone's familiar with, but it's really impressive when you think about it, because that's a lot of the terrestrial surface, right? And if we're going to protect animals and ensure that they can persist, we need to make sure that they can still use some of this 40%. Okay. Oh, my screen won't advance. There it is. Okay. So I'm going to talk to you about today a little bit about biological corridors, but very briefly, because I know that I imagine many of you go to like several of these talks this summer, and raise your actual physical hand if you saw David Miller's talk a few weeks ago about Algonquin to Adirondack's biological corridor. Okay, I can't actually see everybody, so maybe that question was at least 3 people I saw. Okay, so for those of you who haven't heard of biological corridors, I'm going to give you a quick rundown of what they are. So what biological corridors are, is they're based on this concept that, okay, landscapes look like this now, right? Many landscapes don't look like that continuous forest. But we can't change everything back to continuous forest. We have we have about 7.6 billion people on the planet. We need to farm to grow food, and we need places to live. So how can we retain these urban areas and agriculture, and also make sure that wildlife and plants can still move? So imagine if you're a farmer, you can see what areas you'd use, but if you were wildlife, there's kind of a little pathway there, but it's a little tough to get through. So biological corridors, this idea has been developed, and I give the definition of the biological corridor here from the Central American Commission for Environment and Development. And this definition says, a geographically defined area which provides connectivity between landscapes, ecosystems, and habitats, natural or modified, and ensures the maintenance of biodiversity and ecological and evolutionary processes. So this idea has been taken up in many parts of the world, and people have really embraced the concept of biological corridors, but it's different everywhere. So for example, this is a picture of the European greenbelt, and you can see the map of its extent. It's huge, right? And in Europe, this is enforced by laws. They're a little different by country, but mostly you can't build on that area. You can't build a road through it. It has higher pollution and contamination protections. So it's a biological corridor with teeth. You can't violate its borders. Here's another famous biological corridor, the Yellowstone to Yukon biological corridor out west in the United States. And this one doesn't have teeth. So you can see in this map that there's all these places where there's protected lands and conservation lands, and they've done some awesome things. Like, I love these pictures of how they help animals cross. This is a bridge that animals, you know, can feel comfortable crossing over the top of. But this doesn't have teeth. And what do I mean by that? If somebody who's a landowner right here decides to sell 1,000 acres of land and build a huge housing development, they can. There's nothing about being in that biological corridor that puts a limit on their land use. So in the US and in Latin America, where I've done a lot of my work, it's more of a concept than a legal entity. So this last example is the Mesoamerican biological corridor. So if any of you have been lucky enough to travel down to Central America to spend time there, it's really beautiful region, and there's a lot of protected areas there, and there's many entities and NGOs working to connect them up so that across many countries you have protected areas that wildlife can travel through. And here's a picture of, like, often we protect riparian areas because, and riparian means at the side of a stream or river, because those are areas that get high traffic by many different taxa. So, okay, this idea is great. It's the biological corridor. Well, we can use the land for agriculture and for houses, but we'll just leave up enough forest or native prairie or whatever the habitat is so that animals can pass through. But it's really important to also ask, does this work for animals, right? Because we can look at that, all of us, and we can say, yeah, I think that understory forest rodent is definitely going to go through here. Can you see my arrow on the screen? You can? Okay, good. So it's definitely going to go through here more than through here. But we don't know that until we test it, right? And that's what field ecology is, going out and testing whether those assumptions are right. Because what's the point of spending a lot of money to protect these lands if it's not actually working? So there's been a lot of, I shouldn't say a lot, there's been some work to see whether corridors are working. And for the rest of the talk, I'm going to explain to you like how the heck you test that, but I just want to show you right here, like an overview. This is a meta-analytic review, so they looked at 78 experiments from 35 different studies. And what they found in this study was that essentially, yes, corridors have a lot of potential. So I'll explain what this graph means over here. It basically means, so these black circles, that's the effect size. So a bigger effect size means it's like the corridor helped more. And then these empty circles is the effect size after they factored in some covariates, factored out the covariates, excuse me. So you don't need to understand all the details, but basically the trend is like, it's kind of what you would expect. So corridors are really helpful if you're a non-avian vertebrate. They're less helpful if you're a bird, because why? Birds just fly. Birds just fly over the whatever's in between the forest patches, right? So the effect size for birds of corridors is just low. They can just fly over the problem. But as you get to less agile organisms, they're less able to move across those things, and corridors are more helpful for them. So if you were at that talk from David Miller, who's the executive director of the Algonquin to Adirondack Biological Corridor, then you're already really familiar with this picture. And even if you didn't go to that talk, you hopefully have heard of the Algonquin to Adirondack collaborative. So I'm on the board of that collaborative. And essentially, this is another biological corridor without teeth. Everyone I saw, except for the person from Boston who's on this talk, everyone else who I know where your town is, you live somewhere in there, to the best of my knowledge. And here's where the Indian River Lakes Conservancy is. So if you're thinking where you are in reference to that, that's where that is right there. So the central part of that corridor is where it's like pink or beige, right? Because animals have a hard time crossing the St. Lawrence River, but through the Thousand Islands region and the Frontenac Arch, there is some movement and it's possible to cross. So that's the corridor. The Indian River Lakes Conservancy lands lie within it, as do many agricultural areas, right? Around this whole region, most of that yellow part is agricultural. So we have this biological corridor that we established because we want to make sure that boreal species in the Adirondacks and boreal species in Algonquin Park can have exchange, especially from the Adirondacks to Algonquin as climate change warms it up down here. And that species can still move across this boundary just as they were able to in the past before humans developed this area. So then let's get to the part of, okay, well, we think that would be nice, but do animals like that idea? Well, you may have heard of Alice the moose. That's not a picture of Alice. I just Googled a moose. Although you probably couldn't, I don't know who among you could tell moose apart, but this is a representation of Alice the moose. And she was like the motivation, she was the inspiration for the Algonquin to Adirondack corridor because back in the 90s at Huntington State Forest that SUNY ESF owns, they radio collared and released a bunch of moose in an effort to reintroduce moose to the Adirondacks. So I'll talk more about moose later, but if you're not aware, moose were hunted out quite a while ago in the Adirondacks during the phase when there were a lot of loggers in the area in the early 1900s, and people basically hunted them out for meat. That's a lot of meat, right? So there were no more moose, but we wanted moose back, so we reintroduced them. And one of them, Alice, went all the way from the Adirondacks up to Algonquin Park with a radio collar on, transmitting her location. So it's just one animal, but we know that this is possible, right? So she was kind of the inspiration for the corridor. But what other species, but is that enough to justify a corridor? What other animals are moving through this area? Well, how can we test if the corridor is working? Any corridor, not just Algonquin to Adirondack. So I think a piece that gets left out a lot is the social effectiveness of the corridor. If landowners in the corridor have no idea that it's a corridor, it's not going to be as effective, right? Because they can decide to do whatever they want on their lands. They're not going to take into account something they're not even aware of. So for this part, for the rest of this piece, I'm going to be telling you like what has been done in terms of seeing how animals are moving through the region. And as I said before, I want you guys to think about what else do you think could be done? What's missing? What species should be looked at? Or is there a different region that should be looked at? Because I'm curious to hear your thoughts at the end. So, okay, there hasn't been a lot of social work on the corridor. There was this paper written in 2004 by some folks at St. Lawrence University, where they basically went around and did like all these surveys of landowners on the US side in the corridor to see if they knew about the corridor and what they thought about it. But that was it, and that's 15 years ago. And our organization, A2A, does conservation action planning workshops on a regular basis, so we spread the word that way. But those have been mostly on the Canadian side because that's where we have funding to do them. They've been all on the Canadian side. We'd like to do some on the US side. So there's social effectiveness. You can test whether people know about the corridor and support it. Ecological effectiveness, there's three main ways I'm going to talk about. So how can you see if animals are moving? You can do camera traps, you can do telemetry or GPS collars, or you can use genetic data. And maybe potentially another fourth one that I didn't really think of is citizen science, which is essentially like, if you've seen a species, you go somewhere and register it. I actually didn't cover that in this talk, although there's one citizen science opportunity I'll tell you about. So I want to tell you, so basically, like, I'm very interested in figuring out what the heck has already been done. And so I've combed through the literature, not just for this presentation, but over the years, seeing what is already known, what empirical studies, like studies where they actually went out and collected data to see how animals were moving. And I'm going to show you literally almost all the ones I've found. There's about, but don't worry, there's not 500. There's about 7. And they all have cool animal pictures. So this was a study that was done, boy, I don't have the date on there, but it was older. I remember it was in the early 2000s maybe. And they were looking, and this was in Georgian Bay, a place I've never been, but you might be familiar with. You can see when I Googled it, that's what came up. Right here, so that's the edge of Lake Huron. And what they were looking at is how are the Masagua rattlesnake, a threatened rattlesnake, how are they moving through part of this region? And they figured out a mitigation method, which is this culvert, if you see that. It's pretty inexpensive to build, and it's built, you can see by the height, that's not going to help a moose go under there, right? But a moose could just walk over the road. It's not a very big road. But for snakes and other small animals, they showed that this actually did reduce mortality. So they went and looked at roadkill along these stretches, and they found that the snakes were using these culverts, and there was less roadkill along those areas. And they used camera traps to see, like, there you see its little tail going in. So that's one kind of small scale study. There's a larger scale project on lynx that was published back in 2012. They weren't actually using camera traps, but I ranked their study in with these other ones because they were using snow tracks, which is another way of just like not actually being there when the animal passes, but finding a sign of it. And they made this really neat study. So you have to look up close there. There's Ontario, and there's a little section that they did. And they looked for snow tracks of lynx across this whole region for 2 years on transects. And then they also measured other things on the transect, like what depth here, what is the land cover type, how much forest cover is there. And then they modeled where there's a high probability of lynx occurring in the region. And so the idea is this is the first step to saying, okay, lynx are actually here. So if we can connect those spots, it looks like they'll actually move between them. And if you're on the US side of the border, there's a camera trap project that's coming up soon. I have on there Erica Barthelmess at SLU. She was working on a project like this, but I don't think has deployed her cameras yet. But there's a guy down at SUNY ESF. Vanessa, do you remember his name? Did you get that email? I just remembered it. It's Paul Jensen.
Paul Jensen.
Yeah.
Yeah.
Thank you.
Yep.
Paul Jensen. So I don't know him, but maybe some of you do. He's proposing to do a large-scale camera trap project across the northern Adirondacks, and well, I want to try and convince him to expand it out to this part of the 8A corridor as well, to go north of the blue line. And so that is looking to be a really interesting project with a lot of collaborators. So maybe we'll get some really awesome data from there. Because as you'll see, as I go through this, there's not a lot of data. We don't have a lot of data. Okay, so the second way is telemetry. And if you're not familiar with telemetry, what that means is trapping animals and putting a collar on them, like these collars that you see here. And then there's 2 different things which I'm lumping under telemetry. One is telemetry, where like you actually have to go near them and triangulate their location with like a radio antenna, and that beeps. So some of you may have done that at some point in your life. I did it recently to find spruce grouse in the Adirondacks. It's pretty fun. But you have to know where those individuals are, right? You can't just start walking in the middle of the mountains. You'll be very inefficient. now that the technology is really advanced, we also have GPS collars. So those are way more expensive, but you can have a collar that actually is sending out a GPS signal all the time. And you don't really have to go in the field that much. You can receive the GPS data on your computer. So the reason I have these 3 species up here is because there has been radio telemetry work done on moose in the Adirondacks, on deer, and then on wolves, but only on the Canadian side of the border, looking at wolf movement. So I don't actually have the data from these 2 studies. I put this citation here in case you're interested in this paper, but Alex French, who I saw get on this call at some point, is on the A2A board as well. And so he was the one who told me about this data, but those studies aren't done yet, so they don't have the data from that. Okay. So finally we get to genetic data, the best way. Why is it the best way? I'll tell you in a minute. So how the heck do you do this? Like, how does genetic data help you figure out how species are moving? Oops, sorry about that. So essentially just a really simplified version of how genetics do this is like, imagine that you're an animal and a major barrier is built through your habitat. In this case, a highway, right? So I took a picture of a highway. I think that's the Northway. I think I put in a picture of Route 87. And that's a porcupine. Why a porcupine? Because raise your hand if you have seen a porcupine dead on the road. Oh gosh, there's so much porcupine roadkill. It's really bad. They just are sitting ducks. So I picked porcupines because roads are a particular barrier for them. But basically, so what can genetics do? Well, if you're using radio telemetry or you're using camera traps, you know where the animals moved in the last couple days, right? Or where they're moving right now with radio telemetry. But you don't know if they're regularly moving there or if that's a stressed individual who's only going to head out there and then die or fail to reproduce. But genetic data can show you long-term, are these animals moving through this area and are they successfully reproducing? Because that's what we actually want, right? We don't want to know like, oh, did the chicken cross the road? We want to know, did the chicken cross the road, meet a rooster, and lay some eggs that hatched and grew up to meet another rooster and lay more eggs? Like, we want to know all of that. And genetic data tells us that. So, okay, we're going from chickens to porcupines, so back to the porcupines. If you could get a genetic sample from this porcupine, and then get a genetic sample from this porcupine, you could look at how many genes were shared between those 2 porcupines and use algorithms to figure out, okay, well, how related are they? And if there's, how related are 2 porcupines separated by this road, and compare that to how related 2 porcupines are that are separated by like the same amount of just forest. Does that make sense? Because you'd think that porcupines just separated by forest are going to be more closely related, right? It's easier for them to stroll over and like find a girlfriend than it is for the ones who live on the other side of this road. So that's like the really basic concept of how genetic data can tell us if corridors are working and if animals are moving. So just to tell you, so I'm not going to talk about any of my work today because I just started doing research up in this area this summer, which if any of you are in academia, you know was not an awesome summer to start research. And I research bats, but I'm just going to mention my work that I did in the past with bats in a biological corridor in Costa Rica, because that's where my genetics expertise comes from. So for my PhD research, I worked down in this biological corridor in Costa Rica. Hopefully some of you have been to Costa Rica. It's a beautiful country. This is the part where this is Nicaragua, and then San Jose is like where you flew into is like down here. So this biological corridor is trying to protect the Braulio Carrillo National Park and Indio Maiz Biological Reserve in Nicaragua. And you can see that over time, it's gotten fragmented, right? The green is forest, there's a key down there, especially by pineapple, which is moving into the region. And if you've never seen how pineapple grows, that's how it grows. It's like the top of a pineapple growing out of the ground, and it's hard to walk through. But bats don't have to walk, they fly, right? So the reason that I picked bats to work with is because I saw them as like one end of the spectrum. I could have worked with birds, but I just like bats better. And I don't like getting up early, and I do like staying up late. So that's pretty much the really scientific reason why I picked bats. So, because I wanted something that flew, because if something that flew was being really fragmented by this development, then things that don't fly almost definitely are, right? So that's why I picked bats. And I'm not going to talk more about this research here, but I did find that big, long distance flying bats didn't have a problem with pineapple, but small understory bats were showing a lot of genetic fragmentation, especially when they were separated by pineapple more than when they were separated by pasture, for example. So, yeah, if you're interested in that, I can talk about that forever, but I'm not going to talk about that today. I'm going to talk about basically just a few things about how we get this data, which is pretty cool, and then some of it some data that we have like this from our region. So, methods of field data collection. How do you get the genes of an animal? Well, you can over here. I guess this is the way that people might think of, which is, this is me, you can't see it very well, but I'm taking a genetic sample from a bat. So you trap the bat in a mist net, then you take a little biopsy punch that's 2 millimeters, and you punch the tail membrane and take a little tissue piece. Now, if that sounds mean, it kind of is mean. It definitely hurts, I would say, but it grows back, it cures over. I would often catch them with like the little hole sealed over and a little white circle, a scar. So it does cure up, but so my work didn't really showcase like one of the great advantages of genetic work, which is that with many methods, you don't have to capture the animals. So you don't have to pester them at all, which if you love animals and wildlife is kind of what you want, right? You don't want like death by scientists for animals. So I put this picture up here. This could definitely risk death by scientists up here, but you can take blood or you can take tissue when you capture an animal. But there's these non-invasive methods, and they were actually, one of the pioneers of these methods was my PhD advisor, Lisette Waits at the University of Idaho. And she had a whole lab dedicated to non-invasive genetic methods. So it was known locally as the poop lab, which is always great to be able to say you're part of the poop lab. And so basically it's not the sewage treatment plant. It was the lab where all the poop of wolves all over Idaho got sent. And we would analyze all of the wolf scat, it's called, to figure out like the pack structure and whether they were reproducing or whether their populations were declining using these methods. So you can see there, that's what it's like to collect poop. Poop is, it's preserved even better when it's cold. But you don't have to bother the wolves. And then here's an example. People have been successful with the craziest stuff for non-invasive genetics. Eggshells and scraping the DNA off the inside of the eggshell. They've successfully amplified DNA from that. Hair snares are really popular. So you can put something that smells good to an animal on a board or something, and the animal will rub against it, and then you can go collect their hair. The weirdest one I've ever seen was following whales around, and when they blow like spit and air out of their blowholes, collecting it and amplifying DNA from that. So people are really creative about this. There's a lot of different ways to do it, but I tried to do non-invasive genetic sampling by going to where bats roosted and collecting like seeds they had chewed up. But I wasn't able to get it to work. The DNA from many individuals was mixed together, as you might imagine. So I captured them. So once you have your sample, this slide might be a little dry unless you joined this talk just because you were interested in genetics, but I'm only going to spend a minute on it. So there's several different genetic markers. Like, you know that we all have DNA, and so you can use, there's 3 commonly used markers. There's mitochondrial DNA, and so that's from your mitochondria, right? Not from your nucleus. It's easier to, it's small, it's much smaller, it's simple, it's easier to obtain, and it lasts a long time. It doesn't degrade as easily. So when I say easy to obtain, I mean like if this poop failed to give you nuclear DNA because it was really old, it would probably still give you mitochondrial DNA. But there's a major catch. It's inherited maternally. So you are not finding out anything about what the males are doing, right? Except for the males whose poop it is or whatever. So you're losing a lot of information, and it's not highly polymorphic. It's highly conserved. It doesn't change a lot between generations because it doesn't recombine, right? It doesn't combine with the father's DNA. It just passes on down. So that's like the easiest, that's like the low-hanging fruit of genetic markers, but honestly, at this point, it's very hard to publish a study that uses only mitochondrial DNA. Microsatellites are kind of the next step in complexity. They're nuclear DNA, so they're from the nucleus. And so they're harder to get from old samples just because they're more complex sections, so it's like more likely that they'll break. And what a microsatellite is, it's just a repeat, like AT, 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 AT in the DNA. It's inherited from both parents, so you can look at males and females, so that's great. It can be highly polymorphic, and that means highly polymorphic means a lot of variety. So like between all of us in this talk, if we looked at one microsatellite, we'd probably, well, this is like obviously a completely uninformed guess, but maybe if you looked at one microsatellite, you might see like 10 different versions of it between all of us, right? Some of us are going to have the same version, and some of us will have different versions. But it's much more likely that we'd find differences in our microsatellites than in a given mitochondrial gene. So the downsides of that is that they're hard to develop, and you usually only get about 10 or 20 of them. So whether or not you're a scientist, you can imagine that, like, well, we've all been living through coronavirus, right? The more data you have, the better decisions you can make. It's the same thing with genetic data. Like, if you're not testing everyone in your population for coronavirus, you don't actually know how many people have coronavirus. The more people you test, the better you can estimate how many people actually have it. It's the same thing for this, right? So the more microsatellites you have, the better, the more confident you can be that what you're saying is correct, but you usually can't get a lot of them. So the last thing that's been used a lot recently is single nucleotide polymorphisms. And that's something that my friend Susan Bailey, who's on this call, who's from Clarkson, I know she uses them a lot in her research. They are just like a single change in the DNA, a single base pair. You inherit them from both parents, and they are getting cheaper and cheaper to develop, and you can find thousands at a time. And so they're very powerful. As you'll see, there's no studies that have been done in this area with SNPs for any animals yet. Okay, so that's your rundown on that. So let's show you the cool stuff of what we have seen. So, wait, I have to ask you something. When I move your heads, do you see your heads moving, or do you just see my screen? Okay, I can lip read you saying, I lip read Andrea saying your screen. Thank you, Andrea. Okay, so this paper was written in 2007, and basically what they were looking at is fissures. So I don't know if any of you have been lucky enough to see a fissure in real life, but fissures are hard to see, but they're fairly widespread, or they were, but then they were hunted out, extirpated in southern Ontario, but then they started to see them again in the late 90s and early 2000s. And so what this study wanted to know is, where did they come from? Where did those fissures, they've been wiped out and they recolonized on their own. From where? And so when you look over here, you can see that it's like, so the shapes that are the same is saying like they are all kind of the same genetic group. So when you look at the new ones in this area, the hypothesis was they probably came from up here. They didn't cross the St. Lawrence, but the genetic data shows that they did. So the new fishers in this area were in the Adirondack genetic group. See that? So not all of them, right? Some of them are their own group, probably from previously or some mixture. But the point is, this is evidence that they're crossing the St. Lawrence. So the fishers can cross the St. Lawrence. Fishers can make that trip that Alice the Moose made if they feel like it. Another species that, this one we don't actually have evidence of them crossing the St. Lawrence, but this is another paper on the pine marten. So a close relative of the fisher, but smaller and cuter. And these guys use microsatellites, and they basically sampled them at all these dots here, which if you orient yourself, like there's Ottawa, right? So this is like middle Ontario. And so they looked at all these sites, and what they found is like there's tons of gene flow. So up there, which the corridor kind of ends around here, but it's doing fine. The pine martens are passing fine through this area. It is effective. So good news on the pine marten, some good news on the fisher. What about lynx? So if this was my class, I'd make you interpret this and I'd cover the results, but I can't cover the results. So I'll just tell you, they did the similar thing to the fishers. They said, let's take genetic samples of lynx on both sides of the St. Lawrence River to see if they're crossing the St. Lawrence River. And the answer is yes, they are. They are crossing the St. Lawrence River. Now, we've never caught them on a camera trap doing that, and we've never radio telemetried them doing that, but we know they're doing it because genetic data is showing that some of these guys over on the Canadian side match with the US DNA and vice versa, right? So that's hard evidence that they are crossing. And then this is the last one. These cute little flying squirrels. This one isn't, I included this because I'm trying to include all genetic papers from this region, but this one is a little different because what they found is that southern flying squirrels are expanding their range in Ontario. And this is interesting because it's like, okay, are species moving northward as climate change warms their habitat? And the answer is likely yes, as we've seen that for a lot of bird species, but here's some evidence for it in squirrels, in flying squirrels. So the southern flying squirrel is hybridizing with the northern flying squirrel as the southern one is able to live further and further north, right? Because it's warming up. So just in summary, to kind of compare the methods, well, let me just sum up there. That's it. That's all the genetic data from the region. So fisher, pine martens, lynx, and squirrels. Now there's some other stuff. There's wolves. I didn't include those because there's some species that they're not really doing in the region that's crossing, or it's just Algonquin wolves, or just species in the Adirondacks. But as far as species that are crossing, that's the data we have. Although I admit I haven't done a literature search since like Christmas. So there might be some new stuff. Okay. So say you're a biologist and you're saying, what should we do to find out what animals are doing in this region? Well, here's a nice comparison chart. So if you're going to use camera traps, it's hard to identify it to individual, but you can do it for some species, right? Exact path, no way. You just know they were caught by your camera trap at that place at that time. Long-term movements known? No, you don't know that. Multi-taxa surveys? Sure. You guys have probably all seen those. If you're on this talk, you probably are one of those people like me that like looks at camera trap photos, looks to see what animals are on there. Sure, you can get more than one animal, more than one species. The cost is moderate. You have to buy all those cameras and put them out there, but you're not really bothering the animals. Telemetry, of course, you can identify it to individual. That's who you put the collar on. You know the exact path they took, but you don't know their long-term movements. These aren't collars that stay on species for like years and years, right? You have to take them off eventually or they fall off. Multi-taxa surveys, not unless you're a billionaire. They're pretty expensive. And they obviously disturb the animal, so you have to capture the animal, sedate it, collar it, and then it has to carry that collar. Genetic data, so extremely high accuracy to individual, right? Exact path known? No, but your long-term movements are known. You can do multi-taxa surveys, you can collect the poop of multiple animals, but it gets expensive. But you're not really bothering the animals unless you end up capturing them like I did. So that's all I have. I wanted to kind of turn it over to you guys now if you have questions, but also to ask you some questions too. So I'll go back to that. I'm going to put it back to the full screen so we can all see each other, but I want to ask you some of these questions, like what species were you hoping to see up there? What species were you wondering if there was genetic connectivity in? And what species have you seen moving? So I'll hand it over to you all. A coyote? Lynn, is it you want to see work on a coyote, or you've seen coyotes moving through your area? Bats, of course, yeah.
I've seen coyote, but I'm just wondering about the initial New York State population that developed about 1950 to 1960. We saw an increase in the number of coyote here, and then tracking where did that population originate from. But you could do the same thing with red fox or any of the major fur-bearing species.
Yeah, for sure. And there was a really interesting paper that I can send Heidi that came out, I want to say like 5 years ago, that did look at wolves in this region to see whether the wolves in Algonquin Park had interbred with coyotes. And they did find that most wolves have a large portion of coyote DNA, and a lot of coyotes have a high proportion of wolf DNA. So when I first moved here and people were calling them coy wolves, I kind of scoffed a little bit, like, that's silly. But then I read that paper and I thought, okay, they're right, you know? So, so that paper does show some about the origins of where they moved from. So I'll send that to Heidi, and I don't know how she can share it, but somehow she can share it with you.
It would be interesting to see moose and whitetail genetic data, I think.
Yeah, Alex, that reminds me, I forgot to mention that. So just like Lynn's question, if you're in the Adirondacks, you know that moose were extirpated and then came back. So they tried to introduce them at Huntington, but it didn't work. Like Alice went really far away and died. The rest of them died. But then they came back themselves. So now we have moose. And they did do genetic work to figure out where they came from. And they didn't come from across the river. They came from Maine and Vermont, based on the genetic data. So I didn't include that because I was looking for papers showing animals moving across that boundary, which some would say, you know, means that it can't be a corridor. But yeah, so there is that data, which I know you know about, Alex, but that was a while ago. Like I read about that in a book that was published in 2005. So the Adirondack Atlas, if any of you have seen that, by Jerry Jenkins. So we could use some new data on that for sure. Anyone else?
Yeah, I have a question. I'm the Boston person, but
okay.
I've spent loads of time over the last 50 years in the Adirondacks. So my question, I hope I'm not falling off the edge here, but as these habitats are being more and more dispersed and you know, stressed, what can you say about the genetic pool of the animals? I mean, as long as they don't have that much habitat, they can't migrate, you know, in the corridors they're accustomed to, and the populations are lower, is there a problem with a smaller genetic pool for them to mate with? And does that make them less viable in the wild ultimately? And, you know, is there a species that we really have to worry about right now?
Yeah, so it sounds like you know something about this because that's like a very excellent question.
No, that's why I'm asking the question.
Oh, okay. Well, so like yes to everything that you said. If you think about, I always tell my students, like, think about inbreeding, like with the European royalty, right? You can see, or I guess sometimes in the US we say with rednecks, but that's not PC. So let's stick with the European royalty. Like, there's all these jokes about inbreeding, right, that people make, but they make those jokes because it's saying like traits show up that because there's two alleles, variants of a gene that are very rare. And so the chances of them coming together, if you're not related, are very slim, the chances of both parents having them. But if both parents are related, the chances of those alleles being the same is higher. And that there's a ton of evidence showing that leads to decreased vigor. Inbreeding has a lot of effects on vigor and the likelihood of successful reproduction. So yes, as populations get smaller and they can't move, that's exactly what happens. They get isolated, they inbreed, and then they could blink out just from the inbreeding, right? Even if they still have their little habitat patch. But the good news is it doesn't take that many individuals to provide like a genetic rescue.
Okay.
And I can't tell you like a number of individuals, but it's pretty small. Like if you ever heard of Isle Royale in Michigan, has anyone ever heard of that place?
Mm-hmm.
So that's a place where there's wolves and moose on an island out in one of the Great Lakes. I forget which lake it is. And the wolves were getting really, really, really inbred because the lake wasn't freezing over. And so there was no movement, gene flow between the mainland and the island. And then like a couple years, the lake froze and one male came over and fathered like all of the next generation's puppies. And so he was more vigorous. And then there was more genetic diversity in the population and they've rebounded from that. Also here in the Adirondacks, there is a project, which I meant to include this in there, and I'm sorry I didn't, spruce grouse. So there's some people at SUNY Potsdam where I work, working on spruce grouse reintroduction to the Adirondacks because we have spruce grouse, but that's what happened to them. Too small, they're inbred. So they're bringing spruce grouse from Canada and translocating them and releasing them to breed with Adirondack ones. But they haven't analyzed the data yet to know if that's working. So that'll be a really cool paper. So we're letting all these guys go, but they don't, like you see a spruce grouse the next year, you're not like, oh, you must be Paul's son. Like you don't know who that is. So they have all this data, years of it, and they haven't analyzed it yet. It's pretty exciting. See if that's working. But.
Thank you.
I also think that loons would be interesting, to take a bird that's so sensitive and needs such clean water, and then through the front neck arch, that maybe there would be more loons there than there are in the more agricultural parts of the North Country. So I've considered that loons might be a nice indicator type species that would be nice to track how they're crossing and where they're crossing.
That's true because they're like habitat specialists, but they can fly. So it makes it more interesting because they have one pro and one con. So it's more of a mystery. Yeah, well, maybe you all could ask Nina about that next week when she's on here. Andrea, I saw that Andrea, are you a teacher? Okay, and where are you based?
Sorry, I'm at Indian River High School in Philadelphia, New York. And so the Indian River watershed is where our district is. So like if there's something that I can get the kids to get involved in, I would love that.
Yeah, so the thing that crosses my mind is, I'll give you my email because this project so one really neat citizen science thing I think for kids would be to put out some camera traps as part of a larger project. And I mean, that data, admittedly, there's tons and tons of pictures with nothing, but when you do catch an animal, it's cool, right? And then you see what's happening in your backyard. And Alex French might actually know more about the availability of, some places you can check out camera traps from the library, but I don't know, Alex, do you know anything about that?
I think Erica got that grant at St. Lawrence University to do that sort of, exactly the sort of citizen science stuff you're talking about through Nature Up North. So I think they have cameras that you could check out and do that with.
Yeah, so, okay, thank you. So I know that was their plan. I just didn't know, you know more than I do if she got.
Yeah, I'm not positive, but I think that's.
But yeah, Andrea, we could connect you. I could connect you with her. She's a professor at SLU, because then the impediment is the cameras. Where are you going to get them? And then Paul Jensen, this person who I mentioned who's starting this project from SUNY ESF, I don't know yet whether he's open to citizen science, because he just emailed a bunch of professors. So
how about the genetic collections? Is there anything that we can do with that? We did some eDNA collections with Cornell's, you know, Cornell's kits. Yeah. And they loved it. It was just it was a great experience. And they just learned a lot about the fact that you don't have to find invasive fish if you find their cells in the water. It was cool. So I just wondered if there's anything more we can do.
yeah, with that, the only thing I would say is, I know the DEC, like their invasive species, or maybe it's the APIP. Somebody remind me what that stands for. It's run by the Nature Conservancy, and it's like the Adirondack Park Invasive Species Program. So it's APIP, and it's run by the Nature Conservancy in Keene Valley, New York. And they have like you can like report invasive species sightings to them. So it's, in terms of providing you with the kits, I don't know of anywhere who would provide you with the kits, but it sounds like you found somewhere at Cornell. But reporting the data, I know that they have some database where they're looking for invasive, because they want to look at how they're spreading, right? Yeah.
Yeah, they looked for populations of fish that should be here and then fish that shouldn't be here. They're looking for both. So are there any other programs besides the eDNA for Cornell, the fish tracker? Is there any, you know, are there any other projects, maybe citizen science, or maybe Erica is the one that I need to ask?
Well, no, I think so. The camera trap projects that are happening around here are Erica Barcelmus and Paul Jensen, who's just launching it, but I don't know if he has a citizen science aspect to it. Mm-hmm. But I can let you know.
Thanks.
I'll just put my email in here and you can. Actually,
thank you.
Yeah, and if anyone has questions like that, you can send them to me. Because I know that they are looking for people to place them. And one of the things with camera traps is you have to get permission to put it somewhere, right? So by pairing with citizen scientists, you can get permission to put them in all different places. So I know they're looking for those partners. Yeah.
You might want to, as far as the APIP, there's actually another PRISM, they're called, Partnership for Regional Invasive Species Management. So there's a St. Lawrence Eastern Lake Ontario PRISM, and you can find that under the DEC. But otherwise, the Nature Conservancy people, they know all those people. I always call the Nature Conservancy people, even though I should call the SLELO PRISM.
You're right. I clearly also always call the Nature Conservancy people, but SLELO PRISM is really active in invasive species. It also has the best acronym ever. The longest possible acronym. Okay, any other questions?
I was just wondering if the photo that you showed of the European corridor was really interesting, and if you could expound on any of the history of how that came about.
I didn't hear the first part of that. Could you repeat it?
The photo that you showed of the European corridor and how it has teeth, how did that come about? Do you know?
I don't know. It does seem like Europe has better luck with passing progressive environmental measures than we do, like not just in our current administration in the US, but historically. So, but I don't know the history of that, except for the fact that it exists and some basic like punitive measures were enacted across countries through the European Union. But yeah, that would be interesting to, so that makes it more effective, right?
Mm-hmm. It's quite the visual. Very cool photo. Does anyone else have any other questions for Kate?
I had one. The greenbelt made me think of, can you hear me okay? The greenbelt that you mentioned in Europe made me think of our own Adirondack parks and the Moose River Plains. And as my understanding is, that's considered forever wild, that construction and improvements can't be made there. It's mainly maintained for both the flora and fauna for the continuation of interruption for the growth there. Is that true? Is it still forever wild?
You're talking about sections of the Adirondack Park itself?
There's an area called Moose River Plains, and there's a road that runs through it. It's allowed for use for recreation, camping and fishing and hiking, and I believe some hunting. So I was just curious that I know that there's no structures allowed to be built. There is no agriculture. It is considered forever wild. And I know there had been some talk about changing that, but when you showed your picture of the greenbelt, it made me think of the Moose River Plains because that's a protected area. And so everything thrives there, and there's a lot of moose activity there, among other things. So I was just curious, but that's the exact example that we need here in the United States. A lot of those areas, a lot of land should be protected to create that greenbelt situation throughout the United States.
Yeah, I agree. And as far as the forever wild designation, I believe that once that's in the management plan for an area, it's very hard to change. Like, it can be changed like anything, but I just remember when I first moved here, there were a bunch of hearings on the Boreas Ponds wilderness. Does anyone remember those? They were trying to decide what designations to give different parts of the Boreas Ponds wilderness, and a lot of people wanted it to be designated forever wild in its entirety. They didn't win. Some parts were designated. What's the thing down from wilderness? It's like wild forest or something, but then you can build roads and things in it, but not houses. All these designated. And so they can be changed, but it's like a lengthy process of gathering public input and doing all these different things. So I'd say it's pretty safe once it's done.
All right, thank you very much. And it was a wonderful presentation. Thank you.
I'm glad you liked it.
I did. Oh, and I'll add, we have a fisher around here.
Oh, lucky you.
Very elusive. He's scary though. He's elusive, and we know when he's been about because we lose 3 or 4 bunnies at a time. He likes our bunnies.
I bet he does.
So yeah, they're very interesting.
I would love to see one. I never have, but
They're quick.
you should get a camera at your property since you see them all the time.
We've had a couple of game cameras here. Mainly we capture the white-tailed deer. We've had coyotes, fox, the fisher. That's how we knew that we had a fisher for sure.
Okay.
I often thought it was a cat, but cats have a nice round body and that long tail and a small head. The fisher is like 3 equal parts. So when you see it and he's moving, that's how we detected it was him and not the woodchucks in the back. So.
Okay.
And we live like 5 minutes from downtown city in central New York. And because we have a lot of untouched area here, all the animals are just, there's quite a few.
Yeah, that's a great example of the fact that animals don't need it to be like perfect wilderness to be able to move through it and use it in some way.
No, we have 2 acres untouched, and it's proof that they flock here. It's like a safe haven. That's the joke.
Except for your bunnies. Okay, well, thank you all for coming. I appreciate you coming out when you can't be in that beautiful building that Indian River Lakes Conservancy has, but rather on the computer. So thank you for joining us. It was nice.
Thank you, Kate, so much for a wonderful presentation.
Thank you, and Andrea, send me an email. Okay, bye.
Thank you all for attending.
Thank
Have
you.
a wonderful evening, guys.
Thank you.
Bye.